Thanks, Blakely, and thanks everyone for joining us today. So good afternoon or good morning or good evening, depending on where you're joining us from. So welcome to this Validate seminar today. As I think many already know, having joined, sorry, I'm hearing some feedback. I assume everyone, Blakely, can you just confirm that you're hearing me okay?
I can hear you perfectly. I will try and mute people, but if I can ask people to make sure they're on mute.
Okay, thanks. Just to say a few quick words about Validate for the benefit of any non-members who've joined us today. Validate is an international network of researchers who are focused on developing vaccines against globally significant diseases caused by complex intracellular pathogens. So we have a particular focus on tuberculosis, leishmaniasis, melioidosis, and leprosy. And Blakely will be dropping some links into the chat for those who might like to find out more about the network. So today marks the last of sorry, I think you muted me as well.
I'm sorry about that. I did, sorry.
That's fine. I hope I was just saying that you'll drop information in the chat for people about Validate. And then saying that today is the last of Validate industry seminars, and we'll be hearing, as Blakely has mentioned, from Oxford Immunotec, who are a global diagnostics company offering regulated ELISPOT assays for T-cell measurements. And I'm very pleased to be able to introduce our speaker, Dr. Yupei Xiao. Dr. Xiao has a PhD in respiratory medicine with over 10 years of clinical research experience. She's also an expert in clinical and scientific communications, so I'm sure this is going to be a great talk. She is the Medical Science Liaison for the EU and rest of the world at Oxford Immunotec. And so without anything more from me, welcome Dr. Xiao, over to you. Thanks.
Hello, is everybody hear me okay?
Yes, and I can see your slide.
Yeah, great. Okay, so good afternoon, everyone, and thanks, Sam, for a nice introduction. So I'm Yupei, Medical Science Liaison from Oxford Immunotec, and thanks for joining us today. So today we will look at tuberculosis infection and the T-cell mediated immunity, as well as the current available clinical tests for detection of T-cell response to TB infection. So as we all know that despite the availability of TB vaccine and also the effective antibiotics, there are still over 10 million new TB cases each year, and approximately 4,000 people die of TB every day. So it's TB still a global, still a top global health emergency and needs our attention more than ever. What is TB? TB is an airborne infectious disease. Mycobacterial tuberculosis, MTB, is a responsible pathogen. Similar to COVID-19, MTB is aerosol bacteria, can be transmitted from adult with pulmonary active TB disease from to another person, while this adult is coughing, singing, and shouting. So as little as 10 bacilles may actually cause transmission of TB infection. From a clinical and public health perspective, patients with TB infection are simply categorized into having latent TB infection or having an active TB disease. So 1/4 of the world population is reported to have latent TB infection, and 5 to 10% of those cases progress to active TB disease over a lifetime. Active TB commonly affects lung, causes pulmonary TB with symptoms such as coughing of blood or sputum, pain in the chest, weight loss or fever. But it can also spread to the other side of the body, causing extrapulmonary TB. So usually adult with pulmonary TB is highly transmissible. In fact, TB can present as dynamic spectrum, like latent TB infection and also active TB disease, just to reflect two ends of this spectrum. It could take months to years to progress from latent TB to active TB disease. For example, before progress to active TB, latent TB infection may also be sometimes progress into a subclinical TB disease stage. So subclinical TB are patients with active culture-positive disease but asymptomatic. Clinically, early diagnosis of subclinical TB allows for the timely initiation of the treatment that appears to reduce mortality. So it is of great clinical value if we are able to capture and treat those with the subclinical TB, and or a vaccine can be powerfully stop patient progress to a subclinical active phase will be very useful. Again, the risk of transmission from latent TB infection to active TB disease, as well as the speed of progression, depends largely on the body's immune system. So LTBI is more likely to become active TB disease in the immunocompromised population. After initial exposure to MTB bacteria, the chances of TB infection is around 30% in healthy people. Among those, for people with LTBI, with normal immune system and no risk factor without treatment, the risk of developing active TB disease is about 5% in the first 2 years, and then after around 10% over a lifetime. So for those with weakened immune systems, the risk is much higher, about 1 to 10% risk of TB progression yearly. So current situation is that the global effort on TB control is still heavily focused on fighting against active TB cases in order to stop TB transmission and reduce TB deaths. There is really high needs for developing more effective TB vaccine to prevent active TB disease. Unlike vaccines available for COVID-19, unfortunately, currently, like only BCG vaccine is the only licensed TB vaccine. So BCG vaccine is actually widely used, has around 90% coverage globally. Despite this, there is still 1 person dies of TB every 20 seconds. So BCG vaccine does actually significantly limit dissemination of disease to the other side of body and protect children from severe form of TB, such as TB meningitis. But BCG does not protect well against pulmonary TB. And the efficacy of BCG vaccine against lung disease in adult is actually highly variable, with vaccine efficacy waning over time. The pulmonary TB, especially in adult, is highly transmissible and has the highest mortality and morbidity rates, so we urgently need a more effective vaccine. Talking about vaccine and vaccine development, it is also very important to have tools or tests able to monitor host immune response after vaccination. For example, the test be able to tell us if vaccination provided protection to against the infection or not. Let's just next to have an overview of the current clinical available host immunity test against MTB infection. So there are skin tests and interferon gamma releasing assay IGR tests. Those tests are based mainly testing immune response to either purified protein derivative PPD or to TB-specific antigens from RD1 region of MTB bacteria. So before 2000, TST has been the main, early 2000, TST has been the main screening test for TB infection. but with very low specificity. Some data reported the specificity could be as low as 70%. So that's mainly due to TST cross-react with BCG vaccine and also most environmental non-TB, non-tuberculosis mycobacteria. So using TST test, it is not possible to determine if a BCG vaccine, for example, can prevent acquisition of MTB infection or not, or if only limit progression from latent TB infection to active TB disease. And so IGER test is a new type of blood test. It overcomes those issues with TST test. So they measure T cell release of interferon gamma following stimulation by specific TB antigen from RD1 region, such as ESA6-CFP10 TB antigen. So this ESA6-CFP10 TB antigen derived from RD1 region are not encoded in the genome of any BCG vaccine, and also most non-tuberculosis mycobacteria. So IGER overcomes the cross-reactive issues in TST test, can provide more accurate diagnosis of TB infection. So far, as many studies reported that IGER tests are used in vaccine trials, for example, for testing the vaccine efficacy in prevention of TB infection. So vaccination is definitely an excellent way to protect us against infection. Our current understanding of immune correlation of protection in TB is still insufficient. Despite the potential of B-cell antibodies, neutralizing antibody in some other pathogens, such as vaccine response induced for COVID, and TB somehow is very different. And it is actually widely accepted that MTB specific T cell immunity is crucial for the protection against MTB infection. So a test able to determine the T cell response to MTB infection is essential to rational vaccine development. So next we will look at T cell mediated immunity and the science behind the test. So after MTB bacteria breathes into the lung, reaches the alveolar space, immune cells such as macrophages and dendritic cells first trying to kill, ingest those bacteria and process those bacteria into small piece of TB antigen, and then express those TB antigen on their cell surface. So immune cells carrying a TB antigen then travel to the lymph nodes, where naive T cells are waiting. T cells are then become sensitized and develop into TB antigen-specific T cells. Also went for rapid replication, develop an army of those TB-specific effector, for example, CD4 and CD8 T cells. Those cells and then travel back to the lung where the infection is. So CD8 T cells mainly attack and destroy infected immune cells, and also CD4 T cells mainly secrete cytokines such as TNF-alpha and interferon gamma to signal and recruit more immune cells to help clear or contain the TB bacteria to form the granuloma. So the granuloma integrity is important in the control of TB infection, especially like interferon gamma is crucial for protection against MTB, along with other cytokines can further stimulate, attract more immune cells into the infection site, also active macrophages to kill bacteria. So from the host perspective, the granuloma is a prison with the potential to ward off infection from the rest of the body. And then from the bacteria perspective, it is a growing collection of phagocytic cells to infect and to replicate within. So granuloma can even, for example, protect bacteria from directly exposing to TB drugs or neutralizing antibody produced after vaccine. This is a big challenge for TB treatment, and vaccine, if only focus on inducing antibody, neutralizing antibody, inducing a B cell response, it will be not sufficient. So for TB, it's really important that you have vaccine be able to initiate robust T cell immunity. As I just shown, latent TB infection occurs when granuloma can only control but not able to kill those bacteria. So they wall off present this bacteria in the center of the granuloma. So this individual might actually only have a small amount of TB bacteria leak from time to time. So the low bacterial burden can then be sufficiently managed mainly by CD4 T cells. And if an individual's immune system is weakened, the granuloma maybe then unlock and release the large amount of TB bacteria to the lung. Bacteria grows out of control, resulting in developing of active TB disease. So in that case, high bacterial burden requires both CD4 and CD8 T cells working together to fight against the infection. The main goal of the current vaccine research one of the main goals is also to help to prevent active TB disease transition from latent TB infection. So both CD4 and the CD8 T cells would be very important target for the vaccine research. Apart from CD4 and CD8 T cells, there are also some other immune cells involved. The innate immune response is the first step of immune response. So most people exposed to MTB are capable of self-clearance due to the protection of innate immune response. And also the innate immune response could happen very quick. And the adaptive immune response is normally mediated by B cells and T cells through cellular immunity. For TB, T cell immunity is crucial for the protection against MTB infection. So following initial infection, there is a significant delay in the induction of acquired cellular immune response. And usually it takes up to 2 to 10 weeks. This delay allows for a marked expansion in bacterial population, could potentially lead to active TB disease. So the best way to accelerate the kinetics of acquired cellular response, adaptive immune response, is through vaccination to achieve the quick elimination of MTB. And also from time to time, a small number of TB-specific effector T cells can develop into memory T cells. So once established the memory, this period can be lifelong, lasting years and decades. So I just talked about Iger test. Aurelia could be used to assess the efficacy of vaccine for the prevention of new infection. So what's the science behind this test? So briefly, after MTB infect lung, the immune cells first trying to kill ingest MTB, process it into small peptides, which the peptide is actually of different lengths as well, and then express this peptide on the cell surface, which of course those will include different size of ESA6 CFP10 TB antigen from RD1 region. And then those cells containing, this immune cells expressing ESA6 CFP10 TB antigen will then travel to a lymph node to prime and active naive T cells to develop an army of ESA6 CFP10 specific effector T cells. So those effector T cells and then travel back through peripheral blood and back to the lung, where the infection site is to kill TB bacteria. So if you taking blood from this person, the blood will contain large amount of ESA6-CFP10 specific CD4 and CD8 T cells. So as for ELISPOT test, T-spot test, for example, after blood collection isolation of PBMCs, those PBMCs contain both ESA6-CFP10 specific effector CD4 and CD8 T cells. And then those effector T cells are being placed into the antigen well and can then be activated by a mixture of different lengths of ESA6 and CFP10 antigen in the test antigen well. And when they are when activated, and then they produce interferon gamma. So the test spot count, which is a spot forming a unit SFU in each well, is the footprint of ESA6-CFP10 secreting effector T cells. So this is the science behind ELISPOT IGRA test. So IGRA test can accurately diagnose TB infection. For example, the clinical sensitivity and specificity for the IGRA diagnosing TB infection could be over 99%. So for summary, when somebody becomes infected with MTB, T cells are activated and developed into TB specific T cells, for example, CD4 and CD8 T cells. And in vivo, this person's TB specific effector T cells migrate through peripheral blood to the site of the infection and further secrete interferon gamma. And then interferon gamma will recruit more immune cells to the site of the infection and also activate macrophages to ingest and kill MTB. So in vitro, if we do the test, the infected person have TB specific T cells circulating in their peripheral blood. So after the blood collection, the PBMC separation, those cells secreting interferon gamma when stimulated by TB specific antigens such as ESA6-CFP10. So IGRA test measures host response to MTB infection. So ELISPOT detect interferon gamma releasing effector T cells. So which actually very useful in terms of if you can use it for detection of new TB infection after vaccination. So this is the end of my talk. Thanks for listening. I welcome the questions.
Thanks, Yupei, for that very interesting talk.
Thank you.
I'm just pausing to see if there are any questions in the chat or any hands going up. I can perhaps start off if no one is jumping in.
Yeah.
So one of the questions I had is that, and I think you sort of indirectly said it, what are the TB antigens that are included in your assays?
So it's a ESA6-CFP10 TB antigen.
Okay. And are there any others that are under investigation, or you just focus on that?
Because not yet at the moment. Like we, as ELISPOT, like T-spot TB test, we have been using these two antigen ESA6-CFP10 from RD1 region since 2001. We actually never changing it. So it is a very specific antigen. And again, as you probably aware, there is another IGRA test, which is quantiferin. So they are recently just switched to only ESA6-CFP10 antigen on their new generation quantiferin plus test. Previously, they're actually using 3 antigen. So after many years, they found only ESA6-CFP10 will be more clinical relevant. So they now make their new product into like only using ESA6-CFP10. Obviously, there is also other like TB TB antigen from RD1 region are under investigation. But like this, in order to be able to use clinically, you need to achieve better sensitivity and specificity. Unfortunately, it's no success so far.
Okay. I see there's a question from Vainant person in the chat, and then another one from Ashraf. I'll read out. Vainant, if you want to put your mic on, you're welcome. Otherwise, I can read your question for you.
Yes, please.
Vainant's question is, how do you correct for immune-compromised individuals?
So for those immunocompromised individuals, if you're using, I will mainly discuss the method on using ELISPOT. So it depends, both IGRA test and TST will have like a negatively impact by immunocompromised condition. But like compared to all the tests, ELISPOT have actual advantage, because for example, for HIV patient, if you have a low CD4 T cell count, and for the ELISPOT, what we can do is we taking the blood, and then we do the cell count, and we make sure every test, like very well, we put 250,000 cells into each well. So in that case, the test won't be affected like by the low T cell number. And again, like for those patients, for example, the COVID patient, the severe COVID patient who is receiving corticosteroids for the treatment, if you trying to do LTBI testing in this population, the corticosteroids would potentially suppress T cell function and also decrease T cell number. Obviously, COVID patients, they have decreased T cell number anyway. So this method, by isolating PBMCs and then also have washing steps and the cell counting steps, is like trying the best to eliminate the interfering substance such as corticosteroids effect on the cells, and then incubate the cells overnight with TB antigen also gives the best chance for cells to recover, like especially the T cell function wise. So it will actually have a better test performance in that way. But obviously, either test does actually affected by the immunocompromised condition, as any test, like any condition affect T cell function, potentially will affect either test.
Okay, thanks. And Vainant said thanks for that answer too. Blakely, I'm just going to ask, I'll keep an eye on questions, but you just need to keep an eye on time and give us a shout when we need to wrap up, because I'm seeing a lot of questions pop up now.
Yeah, we've got plenty of time, but yeah, there are a lot of questions to get through there.
So next is from Azrat. I hope I'm pronouncing that correctly. What is the risk of NTB transmission from LTBI in quantitative terms? Is it 0?
Sorry, could you say again?
What is the risk of NTB transmission from latent TB infected individuals?
It depends. For example, I mentioned earlier, for the people with healthy immune system, might be 5 to 10% of risk of developing to active TB during the lifetime, especially for those latest TB contact person. So if this person get contact with active TB disease patient, and then within 2 years, and after the initial contact, they will have around highest like 5% of risk of getting TB infection, the active TB disease. And then for those HIV and also with immunocompromised condition, the risk is varies. It could be much higher, could be 1 to 10% yearly.
I think perhaps the question was more focused on the person with latent TB and how infectious they would be. But that's a very tough one.
Well, you said so. The patient, the person with latent TB, do not actually is not infectious. They won't be able to transmit TB to another person. Again, so for the only risk is actually adult with pulmonary TB, with a TB infection in lung. So if this person coughing, speaking, and stay in the same environment with another person for a certain amount of time, and then this another person will have high risk of getting infection. So only adult in pulmonary TB have a high TB transmission rate.
Okay, thank you. So there's another question from Faria on, can you kindly shed some light on the molecular assays like PCR or gene expert for TB diagnosis?
So gene expert PCR is more of diagnosis of active TB disease. So unfortunately, maybe I can show you on my previous slides. It will be easier for you to look at it.
Bear with me, Yupei. I turned off sharing. Just bear with me.
Okay. Can you see
there we go. Should be fine now.
Yeah. So basically, if you see this two line here, the culture, the imaging, as you could see, the PCR and is
Yupei, you're not actually sharing the screen at the moment, is that? Sorry.
I don't see it.
Let me share again. So I shown you this slide before. If you see here is the culture line is here, and then on y-axis is like a bacterial burden. So for PCR and also a sputum culture is only implies to the patient with confirmed active TB disease already. For those with latent TB infection, the bacterial burden in their lung or in their body are so low. So the PCR and the culture is not sensitive enough to pick it up. So for the latent TB infection diagnosis, so far only TST and IGR test can be used. When you talk about PCR, gene expert, and the sputum culture, it's more to do with diagnosis of active TB disease. I hope this answers your question.
Okay, I am not hearing from that person, so I think that that does cover it. Just as Blakely said, if you wanted to speak your question, you are welcome to say so in the chat, and then we can give you the floor. I'm going to move on because I see there are a lot of questions coming up. Next one is from Hero, and how is the sensitivity of the assay with different clinical forms of TB? And there's another question later on about distinguishing between latent and active TB. So I guess that's a similar question.
Yeah, I would answer the second question first. So this differentiation of latent TB from active TB is actually how to say still a challenge for IGR testing and for TST testing, because so for IGR test and for TST test, for example, it will be able to tell if a person have TB infection or not. It includes latent TB infection and active TB infection, so it's difficult to differentiate. And for the clinical performance, so for example, the product insert, the claim, be able to use clinically, when we're talking about IGR testing, is more to do with detection of latent TB infection, which is the performance of IGR testing having a really good performance. The sensitivity, specificity could be over 98, 99% in terms of clinical use. Yeah.
Okay, thanks. So the next question comes to the use of ESAT6 as an antigen in vaccines under development. So I guess this speaks to parallel development of diagnostics and vaccines. And so the question is, how would you adapt the test to discriminate ESAT6 vaccinated individuals from TB infected?
If the new vaccine comes out, so we also have a well for CFP10. so that is a way to use it. It's more like, in that case, the T-spot TB testing could be more interesting because in a T-spot TB test, so one well, we actually have a mixture length of ESA6 well, and then the second well is the CFP10 well. So if you using a ESA6 vaccine, so when you vaccinated a person, you take the blood from this person should have really high response picked up by the in the ESA6 well. But if this person not actually infected with latent TB or is not a TB patient, and then your CFP10 well should be 0 or should be below the cutoff value.
That's an interesting take. I hadn't thought of it in that way. Then the next question, I think you've kind of answered, and that is, have you tested whether patients always have a sufficient number of MTB-specific T cells in circulation?
You mean the cells, the T cell? So sorry, I don't get it. Could you explain again?
So the question is, have you tested whether patients always have a sufficient number of MTB-specific T cells in circulation?
So MTB-specific T cells in circulation is more to do with stable latent TB infection?
So maybe Nikki, you would like, I don't know if you'd like to go on and clarify your question. Is this about sequestering at site of disease, or perhaps you could explain?
Yeah, I mean, for the ELISPOT, if you have antigen-specific T cells that you're testing in the peptide pool, or in the antigen pool, and you're sampling PBMCs from a patient, do you know whether they have enough T cells that are actually specific to that particular antigen, since not every T cell receptor is going to bind that antigen?
So basically what we do is, after the PBMC isolation, usually, how to say, for a healthy human being, there will be 70 to 80% of cell population will belong to lymphocyte. And then, so we are actually knowing that within this population, we will have enough T cell response. And also inside the test, we actually have a negative control and positive control for well. So what the positive control well, we're actually using a antigen, which is be able to tell if we actually have enough T cell response inside the whole PBMC collection.
Okay, thank you.
And also, how to say, as our, this is a secret, as our vaccine design, our test design. So for each antigen well, CD4, ESA6, CFP10 antigen well. So we actually have the different lengths of peptide. We all know that longer lengths of peptide, for example, ESA6 peptide, will be able to activate mainly CD4 T cells. And then the shorter length of peptide mainly activates CD8 T cells, but there is no clear cut. There is the longer length peptide do actually activate a small proportion of CD8 T cells. So by like alter the length of peptide, we'll be able to capture both like CD4 and CD8 T cells in each antigen well.
Okay, thanks. The next two questions, there's a question about, have you any thoughts about the mechanism for IgG reversion to negative after converting to positive?
Yes, it does happen. So there is many publications on the, for example, TB contact. Initially, they were like IgG actually positive. And then after a couple of years, like within 2 years time, and many of them converted into IgG negative. So it is actually happens because so IgG testing is measures interferon gamma secreting effector T cells. And also from effector T cells develop into the memory T cells. That actually takes a little bit time. It depends on the test, the time point when you do the test. When you do the test, you might just capture the patient having the effector T cells. And then after the infection, and those effector T cells powerful enough to kill the TB bacteria, and then there is no any bacteria inside the body, and then the bacteria, the effector T cells could just die off, and without necessarily developing into any memory T cells. In that case, if you do the second test again, you won't be able to capture any effector T cells, and then there will be negative results. That's actually a good thing.
So I'm getting the signal that I can take one more question. So I apologize for those that I've missed. I'm just going to, sorry for those I'm skipping, but there's one question on, does your test perform equally in TB endemic populations as in non-endemic populations? If no, so go ahead.
Yes, there is a publication, like a T-spot TB test is actually used globally. And yeah, it is from the current clinical data, in the high TB burden country, we actually seen the similar report to what like currently UK does. Yeah.
Okay, thank you, Yupei, and thanks everyone for the enthusiastic questions. It's really great to see this participation and the really good number of people attending the seminar today. I think it speaks to the interest in the topic. So thank you very much. It was lovely to hear from you and meet you virtually.